this. So hello students and welcome back to another video tutorial by Genome Biotech. In this video tutorial, we would be talking about the process of RNA processing. How does RNA processing take place? What is RNA processing and in what manner the RNA processing takes place in a cell? Okay. In my previous lecture, we have already finished motifs. So now there are two antibiotics from which different questions have been asked. And what are those two antibiotics and how the questions have been asked? One is referred to as rifamycin, another one is referred to as actinomycin. Okay. These are the two antibiotics that inhibit the process of transcription. Rifamycin, which inhibits transcription, is semi-synthetic derivatives from rifamycin, okay, a plant product. It will bind to the active site of the RNA polymerase, binds to active site of RNA polymerase to active site of RNA polymerase, active site of RNA polymerase. So the active site of the RNA polymerase is being bounded with the help of rifamycin, which is a semi-synthetic derivative of rifamycin. Rifamycin binds in a pocket of the beta subunit and blocks the pathway of elongation of the RNA. So the RNA elongation is being halted. That is, it is going to bind and inhibit the process of RNA elongation. Next, actinomycin D. Actinomycin D is an anti-cancerous agent. It will prevent the development of cancer. So when you talk about actinomycin D, it binds tightly and specifically to the double-stranded DNA molecule, prevent the double-stranded DNA molecule from being actively template for the RNA synthesis. So again, the process of transcription will be blocked with the help of actinomycin D. Actinomycin D will be inhibiting the process of transcription by blocking the RNA synthesis that would be taking place. Now, where does actinomycin D binds? Actinomycin D intercalates between the adjacent GC base pair. So it is going to intercalate, it is going to move into the adjacent GC base pair and is going to move in between them because it is strongly hydrophobic and its di- uh, dim- it diminishes per dimensions permits it to slip into the spaces between the two nitrogenous bases of the double helix. The two cyclic peptide structure binds to the minor groove of the double helix. Now see, this is very important. Whenever you look at the structure of the DNA, there are two sites which are present over the structure of the DNA. One is referred to as the major groove, another one is referred to as the minor groove. Now, supposingly this is a this helical structure of the DNA that you can find. One end is referred to as 5 prime to 3 prime. Another one is present here, that is 5 prime to 3 prime. These two strands of the DNA are wrapped together in an anti-parallel direction. Okay, so this is 5 prime and this is going to be 3 prime. So when this is 5 prime to 3 prime, what you can find that the two strands are oriented in anti-parallel direction with regard to each other. Here you find that there is a groove that is referred to as the major groove. This is a major groove. Now major groove is the site, major groove is the site for the binding of transcription factor. Here binding of transcription factor will take place. Binding of transcription factor. Transcription factors are going to take place. Then there is another groove that is referred to as the minor groove. At the place of minor groove, the minor groove is empty. This is the minor groove and the minor groove is empty. Groove is empty. Once the minor groove is empty, okay, then this minor groove could be the site where the drugs could bind. Okay, so the DNA binding drugs, cancer drugs, cancer drugs, okay, could be used to target this minor groove. And this is how we are using actinomycin D. Actinomycin is such a drug that is going to be targeting this particular site that is referred to as the minor groove of the DNA. Once it will bind to this particular site, it will inhibit the motion of the RNA polymerase. Okay, so the RNA polymerase will not be able to move. As it will not be able to move, so it would be halted and the transcription process is going to be halted so that no process of transcription takes place, and hence the cell are going to die off, or the cell will not be able to replicate much more faster. Okay, now what we have to start is the next topic, that is referred to as RNA processing. What do you mean by RNA processing? RNA processing is an evolutionary conserved mechanism that is present exclusively in the case of eukaryotes. See, in the case of prokaryotes, there are no introns. So neither introns are to be excised, nor any capping is required, nor any tailing is required in the case of prokaryote. But in the case of eukaryotes, there are three specific processes that are being involved in the process of RNA processing. The three different processes which are being involved are capping, cleavage and polyadenylation, and RNA splicing. First is referred to as capping, second is referred to as cleavage or polyadenylation, and the third one is referred to as RNA splicing. So these are the three different processes which are being involved. So the first process is capping. In this particular process, what we have to study is about the addition of 5 prime cap over the nascent RNA molecule which is being produced. In the next process, we would be talking about the polyadenylation. How does polyadenylation takes place? Which are the proteins which are being involved in the case of polyadenylation? And the third process that we are going to start is about the splicing of the introns. All these three processes, when combined together, they will lead to the process or the phenomena of RNA processing. So let us start with the capping reaction, that how does capping reaction takes place, how does tailing reaction takes place, that is polyadenylation reaction takes place, and how does splicing takes place. In today's lecture, we would be targeting the capping as well as the polyadenylation. Okay, so what do you mean by capping? Firstly, why capping is absent in the case of prokaryotes? In case of prokaryote, co-transcriptional translocation takes place. So there is no need of capping. Okay. So why capping is absent in the case of prokaryote? I'm just writing it out here. You can note it down. See, in case of prokaryotes, in case of prokaryotic cells, no capping or tail or polyadenylation. Polyadenylation is required as as the process of as the process of transcription, as the process of transcription as well as translation simultaneously takes place simultaneously, so that proteins undergoes so that mRNA so that mRNA undergoes translation without fully synthesis. Now, without even fully synthesis, the mRNA is going to undergo translation. So no specific cap is required. Moreover, as introns are absent, so no splicing is involved. And ultimately, as you know that there is no tail region which is present, so there would be no polyadenylation also required, because there is no tail region that is being produced. Tail region is produced towards the end of the reaction, when the entire process of mRNA biosynthesis has taken place. In the case of eukaryotes, the story is a bit different one. In the case of eukaryote, when you find there is a central dogma of molecular biology that operates in the case of eukaryotes, and that speaks out for itself, that how does the formation of this particular protein molecules, how does the formation of mRNA takes place. We know that the DNA molecule which is present within the nucleus will get converted into DNA, will get converted into hnRNA. Okay, it will get converted into the first RNA, that is referred to as hnRNA. hnRNA will get converted into mRNA. How hnRNA is going to get converted into mRNA molecule? First, DNA getting converted into hnRNA, the process is called as transcription. This is transcription. And transcription takes place with the help of the enzyme RNA polymerase. RNA polymerase. Whereas hnRNA gets converted into mRNA with the help of a spliceosome complex. This is the spliceosome complex. And the proteins that are being involved are the snRNA. snRNA, small nuclear ribonucleoproteins, snRNPs. Okay, these are the proteins which are being involved in the processing of this hnRNA into mRNA molecule. Okay, so what we have to do now here from, because this mRNA is all being produced within the nucleus. Okay, now supposingly this is the nucleus of the cell. Now, once the mRNA which is being produced within the nucleus, it has to be transported out of the nucleus into the cytosol. Now, how the mRNA which is going to be biosynthesized in the nucleus is going to be transported. Gene will be leading to the formation of an mRNA. Now this mRNA has to be transported. So the mRNA is going to be translocated. During the process of translocation, it has to be protected at its 5' end as well as at its 3' end. So the 5' end protection is being provided by 5' capping. Protects mRNA from the 5' end. Protects translocating mRNA. Translocating mRNA from 5' end. From 5' end. 5' end. Okay, whereas the 3' end which is present, there should be polyadenylation. Poly-A tail should be added. Large number of adenosine residues must be added. When these large number of adenosine residues must be added, this will lead to the formation of polyadenylation. It will perform different functions. We always know that polyadenylation protects the mRNA from degradation at the 3' end. Okay, so this is being added and it will help in protection of polyadenylation. provides protection to provides protection to mRNA at its 3' end. Okay, so 3' end protection is being provided to the mRNA through the process of polyadenylation. Okay, now eukaryotic mRNA has a peculiar enzymatic appendix caps like structure consisting of 7-methyl guanosine residue, which are being attached via 5' to 5' phosphodiester linkages. Now, during transcription, 7-methyl guanosine is added to the 5' end of the nascent polypeptide chain. Now, what happens during transcription? There is a nucleotide that is being added, that is referred to as 7-methyl guanosine is added to the 5' end of the nascent mRNA molecule. So there you can find that this is the nascent end of the mRNA molecule that contains a nucleotide triphosphate. Now, to this nucleotide triphosphate, a guanine residue is being added. Now, while the guanine residue is going to be added, there is going to be a release of the pyrophosphate molecule. Which pyrophosphate molecule will be released? You can imagine that in this particular reaction, the innermost phosphate is going to be attacked. This phosphate will attack the innermost phosphate of the incoming guanine nucleotide. When it will attack the innermost phosphate of the incoming guanine nucleotide, it will release the pyrophosphate molecules. Two phosphate molecules, beta and gamma, will be released. This is beta, this is gamma. These two pyrophosphates will be released. Once these two pyrophosphate will be released, pyrophosphates will be released. Pyrophosphate will be released, will be released, will be hydrolyzed, hydrolyzed. Pyrophosphates will be hydrolyzed. Once the pyrophosphate will be hydrolyzed, it will produce ample amount of energy so that the reaction can take place. And now the guanine molecules will be present at the first site. Once the guanine molecule are present at the first site, then guanine 7 methyl transferase enzyme will be able to add methyl group at the guanine residue. Then it will add a group that is referred to as the methyl group at the guanine residue. And this will lead to the formation of 7 methyl guanosine residue. Okay, methyl group being added at the guanosine residue, then it will lead to the formation of 7 methyl guanosine. Now, see this. If the methyl group is present only at one of the nucleotide, then it is referred to as cap 0 only when the methyl group is present at the first nucleotide, that is guanine, at the 5' end. This is the 5' guanine. Okay, when only the methyl group is present at the 5' guanine, it is called as cap 0. When it is present both at the 5' guanine as well as at the next nucleotide, then it is commonly referred to as the cap 1. And lastly, the cap 2, when the 5' guanine contains a methyl group as well as the nucleotide, the next nucleotide, the ultimate nucleotide and the penultimate nucleotides, both of these contain the methyl group, then it is said to be cap 2. So cap 0 contain only a single methyl group. You can find here, this is the methyl group that is present in the case of cap 0. Then cap 1 will contain two methyl group. This is the first methyl group, this is the second methyl group. So there are two methyl groups which are present at the cap 1. Then at cap 2, there are two methyl groups which are present. One is here, another one is here, and this is here. So there are three methyl groups, then it would be referred to as cap 2. So there are three different cap regions, cap 0, cap 1, and cap 2 regions. Any doubt anybody has, you can always text me up now only and I will answer your queries. See, the most important question that is being asked, where in the entire biological world there is 5' to 5' linkage between the positively charged 7 methyl guanosine residue. Okay, 5' to 5' linkage is only present at one site in the biological world, and that is at the capping region of the mRNA. Okay, so that is very important. You must note it down. I'm writing a note here. Please do understand this out. See, this is the region where 5' to 5' linkage is present in the biological world, in the entire biological world. 5' to 5' linkage is present only, linkage is present only at the 5' region, at the 5' cap region of the mRNA molecule. Okay, this is important. Please kindly note it out. Such type of questions are being asked. Next, moving now to the polyadenylation section, that how does polyadenylation is going to take place. And now the cap could be cap 0, cap 1, and cap 2. Sir, which is much more effective, cap 0, cap 1, or cap 2? See, the amount of methylation will provide much more stability. The greater the amount of methylation, the greater would be the stability. So just writing a simple formula about it. So the simplest of all formula could be the stability of the mRNA. Stability of the mRNA molecule is directly proportional, the stability of the mRNA molecule is directly proportional to the extent of the methylation present. So what we can say that is that the cap 2 is going to be much more stable as compared to the cap 0. Any doubt? Okay, so this is going to be directly proportional, a very important conclusion that could be drawn. So the stability of cap 2 will be greater than that of cap 1 and will be greater than that of cap 0. Okay, so this is the sequence of the stability of the three different pattern of methylation. Okay. Okay. Now we have to move forward. The next is referred to as polyadenylation. Now, how does polyadenylation takes place? Let us have an animation of the polyadenylation, that what is the effect of polyadenylation and what manner polyadenylation is going to work. So moving firstly towards the process of transcription termination and how does polyadenylation takes place. So I'm just moving towards transcription
Eukaryotic mRNA has to be
termination. Now see this. We always know that the eukaryotic mRNA is going to be terminated at a particular site using different methods. It could be the intrinsic method of termination, that is row independent method, or it could be the row dependent method of termination. Now, what we have to study here is that there are specific proteins which are going to participate in the process of polyadenylation. Those proteins include CPSF, cleavage factors. All these proteins are previously bind to the are previously bound to the carboxy terminal domain of the RNA polymerase second. And later on, when the process of termination is taking place, when the termination site is being reached, then only CPSF and cleavage factors are being transferred from the carboxy terminal domain of the RNA polymerase second over to the mRNA molecule. And then only they will catalyze the process of polyadenylation. How and in what manner? So let us have a look at it. Okay.
to be processed in various ways before being exported from the nucleus for translation. One such process takes place during the termination phase of the transcription cycle, where a long chain of adenines is added to the 3' end of the mRNA transcript. This process is called polyadenylation.
Okay, so polyadenylation refers to the process in which large number of adenine residues are added towards the 3' end of the mRNA or molecule once it has been biosynthesized. This phenomena is called as polyadenylation. Now, which are the proteins involved in polyadenylation and how does polyadenylation takes place, that we have to understand.
When you have completed this exercise, you should understand the process of polyadenylation and understand the proposed models of termination in eukaryotes.
Now, you must understand the process of polyadenylation and what manner polyadenylation is taking place, how the eukaryotic termination is going to take place.
Polyadenylation involves several factors. The extended C-terminal domain of the largest subunit of RNA polymerase, often called the polymerase tail or CTD tail, is involved in recruiting two of the enzymes necessary for polyadenylation, the cleavage and polyadenylation specificity factor and the cleavage stimulation factor.
This is important. Now, you know that the RNA polymerase second, when has reached the site of termination, recruits over its carboxy terminal domain certain proteins, the cleavage and polyadenylation specificity factor, which will specifically cleave and provide the site for the polyadenylation to take place. And its activity is being regulated by cleavage stimulation factor that will activate the activity of cleavage and polyadenylation specificity factor. Now, how the reaction are going to take place, how the polyadenylation is going to take place, let us have a watch.
When RNA polymerase has reached the end of a gene, it encounters a specific sequence called a poly A signal sequence. Once this sequence is transcribed into the RNA, it triggers the transfer of CPSF and CSTF to the RNA.
Now, both the proteins, whether CPSF or CSTF, both these proteins, you should have told me if the screen is not visible. Okay, I don't know why the screen is not visible to you. Uh, Sorry. Is this visible to now all of you? CPSF and CSTF? Okay. So you must know that now here you can find out that there are two proteins which are being involved. CPSF, another one is referred to as CSTF. And these proteins are always present at the carboxy terminal domain of the RNA polymerase second. These proteins which are present at the carboxy terminal domain of the RNA polymerase second will then be translocated over to the nascent hnRNA that is being produced. The CSTF cleavage factor will stimulate the cleavage and polyadenylation specificity factor, and then this cleavage and polyadenylation specificity factor will cleave the nascent hnRNA at a particular point. Then the polymerase signal will be added to it. Okay, so let us have a look that how does this process takes place. See this, it has bounded, it will make a cleavage and then polyadenylation.
Once CPSF and CSTF are bound to the RNA, additional cleavage factors are recruited as well, leading to the cleavage of the RNA.
Now you can find it out that see, cleavage and polyadenylation specificity factor as well as the cleavage stimulation factor, both of these factors are previously present. They are not being introduced, they are previously present within the structure of the RNA polymerase. Both of these factors were a part of the CTD domain. CTD domain. Okay, CTD tail. They were present over the CTD tail, and then they were being transferred to the particular site over the nascent hnRNA, where the CPSF activity was being activated by CSTF. Note the name of CSTF, cleavage stimulating factor. So this is the factor that would be stimulating the cleavage, and cleavage and polyadenylation specificity factor will be the one that will be activated. Once it would be activated, it would be cleaved. Next point. Just a second. Moving forward. And now, this is where the CPSF binds.
An enzyme called poly A polymerase is then recruited to the cleaved RNA. Poly A
Once,
polymerase
see, once the RNA has been cleaved, once the RNA has been lysed at a particular site, okay, it will produce a new site. And to this particular site, another RNA molecule is going to work. See this? This is the fragment that has been removed. It has been cleaved. Now another protein fragment is going to bind, and that fragment will be referred to as the poly A protein that is going to add the polynucleotides to the growing chain. How? Look here.
adds about 200 adenines to the new 3' end produced by the cleavage of the RNA.
Okay.
Poly A polymerase uses ATP as a source of adenine.
So here the poly A polymerase is going to bind. Poly A polymerase will use ATP and will elongate the chain up till 200 to 250 number of nucleotides being added constantly, forming a very long poly A poly A tail. Okay. So the first enzyme was CPSF. Second, CPSF was being activated by cleavage stimulating factor CSTF. Once the cleavage took place, then additional cleavage factors were being involved. Then poly A polymerase catalyzing the addition of large number of nucleotides. Ultimately, Ultimately, when large number of nucleotides were being added by poly A polymerase, it will signal that the transcription, that this is the poly A tail which is present. Look here.
Poly A polymerase utilizes the same mechanism as RNA polymerase. However, it does so without a template. It is not clear what determines the length of the poly A tail, but that process involves other proteins that bind specifically to the poly A sequence.
Now see this. Poly A polymerase as well as poly A binding proteins are involved in elongating the chain, and 200 to 250 adenine residue could be added to the 3 prime end of the tail. So how many number of residues can be added? 200 to 250 at the 3 prime residue. This is the 3 prime residue. There you can add 200 to 250 number of nucleotides. Okay, next.
RNA polymerase does not dissociate immediately when the RNA is cleaved and polyadenylated. Instead, it continues to move along the template, generating a second RNA molecule that can grow as long as several hundred nucleotides before terminating. However, it is clear that the process of polyadenylation is required for termination.
Now, this is the next process that is not shown in Pathfinder, generally not explained in book, except in very standard book such as the book of cell biology, Bruce Albert's The Cell, or in Lodish. Here you find it out that even after the termination of the transcription has taken place, your protein has been made and it is ready to be delivered into the nucleus. This is the protein, this is the mRNA, sorry, this is the mRNA that has been made. The mRNA has been processed. Okay, it is going to undergo processing. Large number of processing is going to take place. We have already seen the capping, that how does capping take place. Capping was taking place by dimeric capping enzyme. Capping was taking place with the help of capping enzyme. Capping enzyme was involved. Okay, so there is the capping enzyme that is being involved. Then after the capping enzyme, the next enzyme that is going to be involved is referred to as the polyadenylation, that is responsible for tailing. Okay, so polyadenylation, adenylation. And the third process is referred to as splicing. But although all these processes are taking place over this mRNA molecule, which is having the cap region, guanine region, methylation at the 7 methyl guanosine, and has large number of poly A tail, everything is going fine. But what happens to the RNA polymerase? Do it dissociate simultaneously? No. Actually, RNA polymerase moves in the forward direction. As it moves in the forward direction, it will synthesize shorter chains of the mRNA, shorter chains of the hnRNA molecule. Okay, these will be biosynthesized by the RNA polymerase. Now, how does these molecules will be biosynthesized and what is their use, that we have to see.
Two basic models have been proposed to explain the link between polyadenylation and termination. The first model proposes that the transfer of CPSF and CSTF from the polymerase CTD tail to the RNA triggers a conformational change in polymerase. This conformational change might reduce the processivity of polymerase, leading to dissociation soon afterward.
Okay, it will change the processivity and will lead to the process of dissociation to take place. Next. Now see this. See, how does process of termination takes place? Termination takes place on the basis of two different models. Transfer of CPSS, SF and CSTF from the carboxy terminal domain triggers RNA changes in its conformation, reducing the processivity of the RNA polymerase over the DNA. Once the processivity is being reduced, the affinity of the RNA polymerase over the DNA is being reduced. Once the affinity is being reduced, RNA polymerase is going to be dissociated from the DNA molecule, and this is the RNA chain that has been released. Moving forward.
The enzymes necessary for 5' capping are displaced during elongation, so the second RNA molecule does not form a 5' cap. The second model for eukaryotic termination states that it is possible that the absence of a 5' cap on the second RNA molecule is sensed by the polymerase, causing the polymerase to dissociate.
The next model that explains the process of termination that takes place in the case of transcription says that because of the formation of a nascent hnRNA chain where there is no cap, as the dimeric capping enzyme is involved in the previous RNA that has been biosynthesized from the structural gene, this uncapped RNA is being recognized. And once this uncapped RNA is being recognized, the process of transcription is being halted and the RNA polymerase is being removed from the DNA molecule, ultimately completing the process of transcription. Now, you will have to apply your knowledge that you have gained just now. Okay, so moving forward, the first question: drag the correct proteins to the carboxy terminal domain. So carboxy terminal domain will have CPSF protein, okay, as well as the CSTF protein. Okay, so this is the cleavage and polyadenylation specificity factor and cleavage stimulating factor proteins that are present, and they would be present towards the carboxy terminal domain of the RNA polymerase second.
Correct.
Moving forward, what causes CPSF and CSTF to transfer to the RNA? How is CPSF and CSTF being transferred to the RNA molecule? Now, binding of additional cleavage factors causes their transformation or transfer to the RNA molecule. Transcription of the poly A signal sequence. Yes, there is a signal sequence which would be transcribed, and that would be referred to as poly A signal sequence. To correctly understand this concept, please look here. I'm again going to share the same with the pathfinder in front of you. See this: whenever polyadenylation takes place. Now listen here. Virtually all eukaryotic mRNA have a series of 200 to 250 adenosine residue at their 3' end, and this end is commonly referred to as the poly A tail that you have already seen. These poly A tails, poly A, are not specified by the DNA and are added to the transcript by a template independent RNA polymerase. So they are being added in the form of a template independent RNA polymerase, which is going to add these nucleotides. And this is referred to as poly A polymerase. Next, the poly A tail appears to have several functions. The poly A tail is responsible for protecting the mature mRNA from the nucleus and it's mediating its export into the cytosol. That is, export of the mature mRNA from the nucleus to the cytosol affects the stability of some mRNA molecules as well as serve as recognition signal for the ribosome. It is the one which would be serving as the recognition signal for the ribosomes. As the mRNA is being transcribed, there is a signal sequence that is being produced. AAU, AAA. It is this AAU, AAA signal sequence that helps in the transfer of CPSF from the RNA polymerase over to the transcript which is being produced. Now, supposingly there is a DNA molecule. Okay. Now, supposingly this is a DNA molecule that is present. See this. And to this DNA molecule, what is being done here? The RNA polymerase has halted. It has a carboxy terminal domain. The CPSF protein, which would be present at this carboxy terminal domain, will be transferred. Okay, it would be having this particular protein that would be called as CPSF. CPSF. Now, what is going to happen? The CPSF is going to be transported to this particular site. Okay, so that is why you are finding CPSF moving at this particular site. This is the site where the CPSF has moved. And CPSF will bind to this particular sequence, which will be referred to as the poly A signal sequence. Okay. In mammals, polyadenylation is directed by a signal sequence in which is present in almost all mRNA and is being referred to as 5' AAU AAA signal sequence. Okay. This sequence is located 10 to 30 nucleotide upstream of the polyadenylation site. It is located upstream of the polyadenylation site, which is often immediately after the dinucleotide sequence CA. There is a dinucleotide sequence CA, and before this dinucleotide sequence CA, this sequence AAUAAA is present. AAUAA is present just before the dinucleotide sequence CA. Here, poly A polymerase will bind, and then there are cleavage factors which are being involved. These cleavage factors will be the one that will be acting and degrading the RNA in between the cytosine and the adenine. Here, the degradation has taken place. Now, it would be this site which would be providing the 3' end for the activity of poly A polymerase. This is poly A polymerase. This is poly A polymerase. Poly A polymerase. When the poly A polymerase will bind, this molecule will lead to the biosynthesis of large number of adenosine chains. And these adenosine chains will be stabilized with the help of large number of other proteins. So there would be other proteins, poly A binding proteins that will be binding to this particular site. Here it is going to bind. This is how binding is going to take place. Now, moving back to our question that we were talking about, are you able to visualize it out? Now, see this. What causes the CPSF and CSTF to transfer to the RNA? The transcription of the poly A signal sequence helps in its transfer to the RNA molecule.
Correct.
Okay, see this. This is the poly A signal sequence which is present. Now, when the poly A signal sequence has been transcribed into the RNA molecule, they will find and move to this particular site and will cleave this particular site. Next question. Would it speak? Given the mRNA sequence below, what is the poly A signal sequence for the mRNA? The pre-mRNA is usually cleaved and polyadenylated at 10 to 15 nucleotide sequence after the poly A signal. You always know that. What is the poly A sequence? You know that the poly A sequence is always AAU, AAA.
Correct.
Okay. You always know this now because see this. See, students, I have already told you what is the poly A signal sequence. The poly A signal sequence is always a conserved 6 nucleotide sequence that is AAU, AAA. This is what the examiner wants to ask from you. Okay. Next question. This, see this. So you can find that there would be large number of repeating subunits that would be present in each and every mRNA transcript because the cleavage and polyadenylation is being triggered by this poly A signal sequence. This will remain conserved. And after this, you will find that there would be residues that would be added. You see this. There are large number of adenine residues being added after the cleavage site. The cleavage site may vary, but the cleavage initiating sequence will remain the same. This is very important. So I'm just writing a note for you all. The cleavage site, the cleavage specific nucleotide can vary can can vary from each other in case of polyadenylation, but the initiating sequence for the polyadenylation is highly conserved. and it is AAU AAA. Okay, this is 3 prime and this would be the 5 prime. Okay, please note it down. This is the poly A signal sequence. Now moving forward to the next question. See this. The distance between the poly A signal sequence and the poly A tail can differ slightly from cell to the cell type. Which of the following could account for these differences? Why you can find that, see this, the distance from the poly A signal sequence, this is the poly A signal sequence which is present, and the process of transcription where this splicing is taking place. Why the distance is greatly varied? You can find that from here you can find that this is the site where polyadenylation is taking place. Here this is the site where polyadenylation is taking place, and here you find that there is a very large amount of polyadenylation, and the polyadenylation site is again varied. Then how the polyadenylation site is being varied, and who varies these polyadenylation site? Okay, so this is referred to as poly A signal sequence. Poly A signal sequence. Okay, just a second. This is poly A signal sequence that is present. Just look here. Which of the following could account for these differences? And what is the difference? The distance between the A signal and the poly A tail greatly differs. Okay, you look here. This is the first distance, and here the distance is just this much. And in the second case, the distance is very large. So why this variation? The additional cleavage factors can vary from cell type to the cell type. This is because of the additional cleavage factor. The mechanism used by polyadenylation polymerase can differ from cell to cell. The RNA polymerase in some cell types lacks a carboxy terminal domain. The poly A binding protein can vary from cell to cell type. Actually, it is always about the cleavage factors, where the cleavage factor is going to vary.
Correct.
Okay, so cleavage factor will ensure how and where the binding is going to take place. Last, mRNA of secreted form, mRNA of membrane bound form, additional mRNA. Now you find here is that there are two different types of mRNA which are being shown to you in the given figure. In the first figure, there is mRNA which is being secreted from the cell. In the second figure, you find that there is an mRNA which is membrane bound, that would be bound to the cell surface membrane. Now, what is the clinical difference in between the two? And try to answer the above question. Okay, secreted. Now, you look here. mRNA which is being secreted out from the cell means that this mRNA is going to be transported out of the cell. Okay, this is the nucleus of the cell that is present. The genetic material is present in it. Now, secreted means it would be mRNA would be biosynthesized here and then secreted out. mRNA secreted out, secreted out. mRNA that will remain bound to the to the membrane. Okay, which of the following could account for this difference? Why there is a difference in between both these types of mRNA? The cells that secrete the antibody have different additional cleavage factors than the cells that produce the membrane bound form. The cells that produce the membrane bound form are unable to properly terminate transcription. The antibody protein is cleaved to produce the secretory form. The cells that secrete the antibody recognizes a different poly A signal sequence earlier in the mRNA. Now, see, this is about the secretion of an antibody. Now, you know that there could be several different types of cells. One antibody producing cells could be the one that could expose an antibody over the surface. Another type of cells could be the one that could be secreting the antibody. Okay. Now, both of these cells, these type of cells, A and B, will differs from each other on the basis of their ability to produce different types of mRNA. So in the first case, you find this is the mRNA. In the next case, you find this is the mRNA. Now the mRNA has to be cleaved. How the mRNA is going to be cleaved? Now see, you cannot have two different types of poly A signal sequence in the case of mRNA, because when you have a poly A signal sequence present, supposingly here, poly A signal sequence, you cannot have two different poly signal sequence, one present here, another present here, so that the cleavage can take place. Because if there would be the poly signal sequence, then where it could cleave here also, or it could be cleaved from here. So the only logical explanation that looks is like this: the cells that secretes the antibody must have additional cleavage factors than the cells that produces the membrane bound form of the antibodies. I don't know, let us check.
Incorrect. You
Okay,
may want
so
to try again.
we should look at the solution. While the two cell types might differ because of different additional cleavage factors, I have already told you this difference alone is not enough to account for such a large difference in the mRNA. Although it is very well known that the two cell types are going to differ from each other on the basis of their difference in cleavage factors. So this is known that the cleavage factors are going to be different, but they are saying that such type of modification will not be able to produce both the different types of mRNA. The cells that produces the secreted form are able to recognize a different poly A sequence earlier in the gene, leading to earlier cleavage, polyadenylation, and termination of transcription. Okay, now I got that particular point. So here what they wanted to say to us is that this is a very specific, highly specific process that takes place in the case of immune system cells, where there are two different types of poly A signal sequence. One poly A signal sequence is present here, poly A signal sequence, and another poly A signal sequence is present here, poly A signal sequence, poly A signal sequence, signal, poly A signal sequence. Okay, so there would be this poly A signal sequence in this case will be protected from cleavage by binding with specific proteins. Those proteins will be able to protect this poly A signal sequence, so that a longer mRNA could be produced. And this additional mRNA will lead to the formation of another part of the protein. And this part of the protein will help to anchor the protein molecule over the transmembrane of the cell. This is the transmembrane part of the cell, and this is the protein that is would be anchored into this transmembrane part of the cell, that is from here. Okay, here this is what has been shown to you in this particular case, the transmembrane part of the protein. Okay, so what we get the answer here is that the cells will
Correct.
secrete and recognize another signal sequence, another poly A signal sequence. Okay,
Polyadenylation is the process whereby
now, please look here. This is the conclusive event. This is the events that are going to take place and again repeat out itself. Okay. So, moving forward, just a second.
a long chain of adenines is added to the 3' end of a eukaryotic mRNA transcript during the termination phase of transcription. Polyadenylation involves several factors. The cleavage and polyadenylation specificity factor, the cleavage stimulation factor, and additional cleavage factors work together to cleave the RNA transcript after transcription of a poly A signal sequence. CPSF then recruits poly A polymerase, which catalyzes polyadenylation.
Okay. So there is the poly A polymerase that will be catalyzing the process of polyadenylation. This is the polyadenylation taking place.
Two basic models have been proposed to explain the link between polyadenylation and termination. In the first model, transfer of CPSF and CSTF from the polymerase CTD tail to the RNA triggers a conformational change in RNA polymerase, leading to dissociation.
Okay. So this will lead to the dissociation, and here the dissociation is going to take place. Next, reducing the processivity. Moving forward.
The second model for eukaryotic termination states that it is possible that the absence of a 5' cap on the second RNA molecule is sensed by the RNA polymerase, causing the polymerase to dissociate.
This is the polymerase that has been dissociated. Next.
You have completed this exercise.
This is how the exercise has been completed. Thank you. Over and out. That's all for this process of polyadenylation. Now, moving back to the Pathfinder, just wait for a second. Now, this is how the process of polyadenylation is completed. The poly polymerase has no specific catalytic activity. The polyadenylation reaction passes through the two-stage. Rather, first, a rather short oligonucleotide sequences is added to the 3' end. There is a short oligonucleotide sequence is added, and the last one, the last molecule that is being added in the second phase, the oligo tail is extended to the full 200 residue length. The reaction requires another stimulation factor. There is another stimulation factor that recognizes the oligo a tail, and that factor is referred to as polyadenylation binding protein. There is the protein that is present called as polyadenylation binding protein, which is present. Polyadenylation takes place from this protein. The poly tail is synthesized and appears to play a role in maintenance of the tail after synthesis. Then the polyadenylation is going to be influenced, and the poly tail is going to play its role. Now, histone proteins. You always know that there are certain proteins which are called as histone proteins. Histone genes are usually among eukaryotic genes in that they do not have any introns. They do not have any poly A tail. Furthermore, in almost all eukaryotes, genes are arranged in multiple tandem repeats, each carrying a copy of 5 histone genes. Suggest an explanation from these features. Firstly, because the histones are a type of housekeeping genes. These are housekeeping genes. Housekeeping genes. As these are housekeeping genes which are present. Okay, it is visible or not? Okay. These are the housekeeping genes which are present. When these are housekeeping genes which are present, then this histone has to be produced in a very large amount. As these are to be produced in a very large amount, so they do not require any polyetail. The absence of introns, look here, the absence of introns and polyetail means that the two of the major steps involved in the processing of most eukaryotic genes are avoided, so that histone mRNA can be delivered rapidly to the cytoplasm. The histone mRNA can be produced much more effectively, and as soon as this mRNA is being produced, this could be effectively delivered into the cytoplasm, into the cytosol, where the translation of this mRNA produced can take place into a protein molecule. Next question. Suggest a means by which you could separate mRNA from other different types of RNA in a eukaryotic cell. The mRNA can be separated from other different types of RNA using an oligo DT or an oligo U sequence to insert inert support to create an affinity column. An affinity column could be created, and in this affinity column, what could be done? A column could be created. In this affinity column, you are going to use beads, and those beads that are going to be used in this affinity column, those beads will have oligo DT molecules attached. These beads will have oligo DT, oligo DT nucleotides present. This will bind to the poly A tail of a particular protein molecule. This is a protein molecule. It will bind to the poly A tail. A, and A. As it will bind, it will be halted in this particular column. It would be halted in this particular column. And then what is going to happen? Then the mRNA could be separated from other different RNA, could be separated, could be effectively separated, could be effectively separated. Any doubt, any problem? That's all for today. We will start with intron splicing. If you had any question, any query, you can just ask me. Thank you. Over and out. That's all. Good night.